Hi everyone, my name is Alexis Fulton, and our topic this week is community structure with focus on diversity and abiotic factors. The paper I've chosen is Additive Partitioning of Plant Diversity with Respect to Grassland Management Regime, Fertilization, and Abiotic Factors. The authors are Klimek, Marini, Hoffman, and Eiselstein, and the paper was published in 2008 in Basic and Applied Ecology. The authors of this paper really wanted to tackle the idea that low to moderate disturbance in grassland areas caused by grazing animals promotes species richness because of the heterogeneity in soil structure and the reallocation of nutrients that it leads to. But they wanted to couple that with the fact that vascular plants diversity in grasslands is also related to abiotic environmental factors. They noted that as species distributions are the result of processes operating at both local and regional spatial scales, it was really important to quantify the diversity of species at various scales and with different factors playing into those diversity outcomes as well. So they came into this research with two questions that they really wanted to answer. The first question was, does species richness among grassland parcels play an important role for total observed regional species richness? And do the relative contributions of additive diversity components to that regional species richness differ among all common and infrequent plant species? And then the second question they wanted to answer is, are the observed patterns of local species diversity components determined by management regime, fertilization, and abiotic factors? The location for the data collection is in southern Lower Saxony in Germany. And it was approximately 1,260 square kilometers. That's primarily used for agricultural land. There are also large contiguous forest-covered areas in the hilly regions of this part of Germany. The average annual temperature is around 8.7 degrees Celsius, and precipitation averages 645 millimeters. There's a lot of fragmentation where there's agriculturally improved but species poor grassland in the lowland areas. And then there are a lot of semi-natural species rich grassland areas that don't have agricultural improvement in the steeper areas in the countryside. Essentially, the authors are defining two different types of managed lands. One of them is called a meadow, and the other they're calling pastures. The parcels are the larger sections of land, and they measured out 60 parcels on 26 different farms. There were 30 that were defined as meadows, and meadows were cut an average of 3 times per year. And then there were another 30 that were called pastures, and they are only grazed. They don't get cut. And within these parcels, they found, they located different plots of vegetation, 3 plots of vegetation in each parcel. The plots were circular and measured 12.6 meters squared, so a radius of 2 meters. And they were all sampled before the first cut or the beginning of the grazing period, depending on the type of management. Okay, so for each grassland parcel to determine the management practice, they had to talk to the farmers. So the researchers used a standardized survey or questionnaire, and they used averages from their responses to figure out that the meadows were cut 3 times on average per year, and the fertilizers used tended to be mineral fertilizers between the cuts. And the pastures, as opposed to the meadows, the pastures were fertilized with manure and liquid manure, so farmyard manure, solid and liquid manure. And mineral fertilizers were very rarely applied to the pasture plots or parcels. The grazing intensity for the pasture land was estimated and then standardized uh, based on were converted to a standard livestock unit. And it was found that the pastures used rotational cattle grazing, and the mean density was less than 1 livestock unit per hectare per year. And a livestock unit was 500 kilograms of life weight, of bovine life weight. Um, uh, they also totaled the amount of fertilizers um, by summing up the organic and the mineral fertilizer that was used, and they used it as a surrogate for management intensity in this study. For the abiotic environmental variables, they derived those using high intensity digital elevation model called DEM, that had a cell size of 12.5 meters. And then they used soil data from vector-based soil maps, as well as a GIS to calculate the slope angle. So the soil quality was based on the digital soil evaluation and vector-based digital soil maps. And then they used a predictor variable to describe the productiveness and fertility of agricultural land based on that soil type for the parcels. Okay, so the sampling design was a nested sampling design, and it was hierarchical. So they took larger parcels of either meadow or pasture. They had 30 from each, and then they sampled from 3 plots from every parcel, giving them a total of 180 plots for sampling. There were 90 plots from the meadow type and 90 plots from the pasture type of management. Those plots within parcels made up the local scale, and then the parcels, as they compared to each other, made up the regional scale. They used a program called partition to determine the average components within and among the parcels to compare the plots at the local scale. And they defined the alpha diversity as the average within grassland parcel components, and then the beta was the among vegetation plot component, and then they got the gamma diversity at the local scale. They found it to be the same as the total alpha, and then for the regional scale, they used the alpha diversity at the local scale and the beta diversities at the local and regional scale sum. And that gave them the gamma diversity for the regional scale. The beta diversity and gamma diversity components were expressed using the same units of richness, and so their contributions to the gamma diversity could be directly compared. They did apply separate analysis for the common and what they're calling infrequent species, which are the more rare species, using the additive partitioning approach. And they defined those uncommon or infrequent species as occurring in less than or equal to 5% of the 60 parcels. And then they were common if they were in at least 25% of the parcels. Okay, the statistical analysis for this study was really a large part of what they were explaining in the paper. The first thing they did was analysis of covariance, or ANCOVA, which compares a factor and a continuous independent variable. And they were trying to test the main and first order interaction effects of the management regime meadow versus pasture on the alpha, beta, and gamma diversity components at the local scale. So they did that, and they adjusted for the covariance of nitrogen fertilization, soil quality, and slope angle. They ran this under the SPSS routine with a type 3 sum of squares. For the local species diversity components, they did a backward stepwise selection procedure to get the most appropriate models. They used scatter plots to check the ANCOVA assumptions for linearity of the relationships. And they verified the distribution of errors and the homogeneity of variances to make sure that the statistical assumptions of the ANCOVA were correct, were fitted. To normalize the residuals, the covariate soil quality was log transformed. And they found that the 3 covariates, so the fertilization, soil quality, the nitrogen specifically, fertilization, the soil quality, and slope angle were not highly correlated with the factor or with each other. So then they checked the spatial autocorrelation in the species composition data with the mantle test to relate the matrices between grassland parcels to a matrix that showed the geographic distances between the parcels. And they found that they found the floristic distances between the parcels based on the presence or absence of species. And they used the Jacquard similarity index for that, which is used to gauge the similarity and diversity of sample sets. They also generated mantle tests for correlation, non-parametric correlation between plant communities and distance matrices for all the grassland parcels and for each management regime separately. And they used the Spearman rank correlations, which assess how well relationships between 2 variables can be described using monotonic function. They did all of this with the R which is a software for statistical analysis provided by our development core team. Then they came up with the mantle statistics supporting the null hypothesis that no significant spatial autocorrelation was present between plant communities and distance matrices for all grassland parcels and for each regime. And they did these all separately. So the results. You can see in figure 1, this is a comparison of the regional diversity of all plants, the common species, and then the infrequent species using that additive partitioning methods. This is again the regional scale, so it's the 30 meadows and 30 pastures. The top line shows the gamma diversity, and then the percentages that are the sum of that beta diversity and the alpha diversity are above on each bar. What they came up with is that there were 190 vascular plant species recorded in the parcels. 149 were dicotyledonous, and 41 were monocotyledonous. In the meadows, there were 120 species recorded, and 30 of them were unique to this type of management. 160 species were recorded in pastures, 70 of which were unique to the pasture type of management. At the local scale, the species diversity was found to be related to nitrogen fertilizer applied. The beta diversity at the local scale was related to management regime and soil quality. The response to slope angle for gamma diversity was significantly different between management regimes. So at the regional scale, there were 35 common species identified. The ANCOVA showed a non-significant influence of management regime, and alpha diversity was negatively related to the amount of nitrogen fertilizer applied. 46 infrequent species were identified in meadows, and 86 infrequent species were identified in pastures. Unlike the local scale, the infrequent species contributed equally to total observed regional diversity. Alpha diversity was negatively related to soil quality, but not significantly impacted by applied nitrogen fertilizers, while beta and gamma diversity responded negatively to nitrogen fertilizer. Overall, additive diversity partitioning was found to be beneficial in providing understanding of local regional patterns of species diversity and spatial scale dependence of diversity components. The relative contributions of alpha and beta diversity components were observed to be consistent between management regimes, indicating that scale-specific diversity components are important. The findings indicate that species richness is probably due to environmental heterogeneity. Both overall diversity and the diversity of rare species were determined by diversity components at the larger scales. The species diversity was not found to be lowered by homogenizing effects such as migration or dispersal, which allows for the movement of species between spatial units. On this slide, we can see some scatter plots showing the linear regressions of the plants diversity in both the meadow and the pasture type parcels. And they clearly show that for the nitrogen fertilization, the diversity decreases with the increase of the nitrogen fertilizer. And then interestingly, with the slope angle, it actually does not make a lot of difference in the meadow type parcel, whether the slope is increased or not, but it makes a kind of a considerable difference for the pasture type parcel. The pastures are the filled in circles, and then the meadows are the empty circles. So you can see on the dashed line down here is showing the meadow line. And it's almost flat, but the solid line showing the pasture, the pasture response to the increased slope angle, the diversity increases noticeably. It was noted that grazing animals in low to moderate density cause small disturbance that lead to heterogeneity and promote species richness. They increase the role of competition at the local scale and encourage the establishment of plant communities from a regional species pool. As opposed to grazing, mowing is non-selective and it actually reduces the heterogeneity. And then locally extreme conditions like steep slopes and low nutrient soils are important for rare plants in particular because it gives them a kind of a leg up. The steep slopes are more resistant to invasion by competitive species than those on flatter ground because there's a phosphorus limitation in the shallow soil that exists in steep conditions. And in this particular study, that the same thing may be true for managed grasslands, for other managed grasslands. All right, unsurprisingly, conclusions were in general that more heterogeneity in land usage and environmental conditions leads to increased diversity. The use of additive diversity partitioning also was found to be really useful in the comparison and quantification of alpha and beta diversity components, contributing to regional diversity in differing grassland management regimes. So the use of the additive diversity partitioning was beneficial, and the study found that different types of land usage and environmental condition increases the number of types of species that will be present in a particular area. The authors did note that there were limitations to their study because other factors were not considered, things like landscape composition, dispersal limitation, and sampling efficiency might be changed, improved, or improved upon. I think that this study has a lot of potential applications in agriculture and in managing for farmers in particular, how they're making use of their land to ensure that they are keeping it as productive as possible, at the same time trying to be environmentally conscious.